Insulin Signaling Pathway. Insulin. Helping glucose metabolism also aid in cell survival mechanisms. After a meal, glucose level increase in the blood act. As in signal, high glucose stimulates the pancreas to release glucose from beta cells. Insulin receptors, which are specific tyrosine receptor, present on the plasma membrane. Insulin receptor contain 2 alpha subunits and 2 beta subunits. Alpha subunits present outside the plasma membrane act as an attaching site for insulin. The beta chains present inside the cell have protein kinase activity, which mean they transfers a phosphoryl group from ATP to hydroxyl group of tyrosine residues in specific target proteins. Insulin binds to the receptor. As a result, the receptors come in close proximity, cause the phosphorylation of beta chains at specific tyrosine residues, which initiate the autophosphorylation of various target proteins. At first, insulin receptor substrate, IRS1, is being phosphorylated. Then it initiates 2 pathways by phosphorylating 2 target proteins. Let's first discuss the metabolic pathway. To store glucose in the form of glycogen, IRS1 activate phosphoinositide 3 kinase, refer as PI 3 kinase, by binding to its SH2 domain. This activated PI 3 kinase converts the phosphatidyl inositol 4,5-bisphosphate, also known as PIP2, into phosphatidyl inositol 3,4,5-triphosphate, PIP3. PIP3 creates docking sites on the plasma membrane to which PDK1 and PKB attaches. PDK1 also phosphorylate PKB. Turn phosphorylate PKB, also known as AKT. Inside the cell, GLUT4 vesicles are present that need to be transported to the plasma membrane, but its movement is inhibited by AS160 PKB phosphorylate, AS160, and allow the movement of GLUT4 vesicles towards the plasma membrane, where they form channels and uptake glucose from blood, thereby reduce blood glucose levels. GSK3 inhibit glycogen synthase by phosphorylation. PKB inhibit GSK3 by attaching a phosphate group. As a result, GSK3 unable to inactive GS, and active GS converts glucose into glycogen for storage. AKT also activate mTORC1, that inactive IRS1 by removing phosphate groups, initiate feedback mechanism. mTORC also has role in lipogenesis, the metabolic process of converting simple sugars and glycerol into fat. Now we see insulin role in gene expression IRS1. After phosphorylation binds to GRB2 at its SH2 domain. It also contains a second domain, SH3 domain, that binds to proline rich of SOS residues, recruit to growing receptor chain. SOS play role in replacing GDP by GTP on RAS. GTP-bound RAS activate RAF1, which phosphorylate MEK on 2 serine residues. MEK activate ERK by phosphorylating it on threonine and tyrosine residues. ERK moves into the nucleus there. Phosphorylate, ELK1, a nuclear transcription factors. Phosphorylated ELK1 joins SRF and stimulates transcription and translation of gene required for cell survival and division.